So it is my pleasure to introduce Łucja Kowalewska. First, I will give a brief introduction or a brief biography. So Łucja got her PhD title from the University of Warsaw in 2015. Then shortly after that, she started as an assistant professor in the same university. And then in 2019, she was visiting, I think, Copenhagen University to expand her knowledge on microscopy and also chloroplast ultrastructure and many different things, but based on electron tomography and TEM. And then since then, she has been combining her expertise in 2D and 3D ultrastructural studies of plastids, and of course, its membranes. And today she's going to talk about it. And yeah, the stage is yours, Łucja. So please go ahead.
Thank you, Juan. Thank you for this kind introduction and also for the invitation. I'm really happy to talk about our science, particularly about the cubic and lamellar configurations of plant plastid membranes and how they can transform into each other, so about the structural dynamics. Let me start with the main, the center of our interest, which is the membrane, the lipid bilayer, the biological membrane. And we are particularly interested in the shapes that this membrane can adopt. This is always connected with the composition of the membrane. So first of all, the shape can be influenced by the shape of the lipid molecules building the lipid bilayer. But all biological membranes are always the protein-lipid mixtures. So therefore, structural proteins can also influence membrane shape. We cannot omit small amphiphilic molecules that can be inserted directly in the lipid bilayer, such as carotenoids or polyprenols, and they can also significantly influence membrane structure. And finally, last but not least, the aqueous surrounding, which is also crucial for the formation of the membrane shape. So biological membranes can adopt several different structural configurations, from simple vesicles through flattened vesicles, characteristic for example, thylakoid membranes in chloroplasts, and also tubules. And such tubules are most probably the precursor structures for the most complicated membrane arrangements known in nature, so-called cubic membranes, which are in fact the configurations of the biocontinuous membranes based on the template of mathematically well-defined strictly periodic minimal surfaces. The example of such membranes is also known in Plastid. It is called Prolamellar body. And I will speak about this structure more later. But I would like to stress that such cubic membranes are not only present in plants, but they can be found in almost all kingdoms of nature, in all eukaryotes, from protista to mammals. And they can be formed from several different types of membranes, from plasma lemma, endoplasmic reticulum, through inner membranes of semi-autonomic organelles, and even nucleus envelopes. Here you can see 3 examples. In amoeba, membranes of the mitochondria during the starvation of this organism. In tree shrew, the retinal cells, the ER of retinal cells of this organism. And in Chlorocebus, in monkey, this is the example of epithelial kidney cells infected by SARS-CoV virus, and the ER then forms the cubic arrangement. So we use plastids as a model to study all of these different types of membrane configurations, and we are happy because plastids can adopt all of these shapes built by their inner membranes. And plastid group is a very divergent group of organelles that are characteristic for a particular organ and also for a particular stage of plant development. They can easily interconvert into each other, which is both pro and con for the studies that we are performing. From the ultrastructural point of view, the most interesting are these two that you are seeing now. So the etioplast, the precursor of the chloroplast, and the chloroplast itself. Because in the etioplast, we can see the typical cubic arrangement, and chloroplast is based on the lamellar arrangements of the thylakoid network. Etioplast is developing in precursor tissues of photosynthetic tissues. So in true leaves and cotyledons, when the initial steps of seedling development occur beneath the soil surface, so without light. So when the light is switched off, so there is no light that reaches the seedling, inside the etioplast, the characteristic structure of the prolamellar body, PLB, is formed. It is a highly periodic structure built from repetitive units, and these units are obviously built by membrane, which is our main interest. This membrane is, of course, separates the space of the etioplast into two completely separated different water channels, one that is connected to the stroma of the etioplast, and the smaller second one, which is a lumen, the same lumen that will be present inside the thylakoids of fully developed chloroplasts. Of course, this membrane is not only built by lipids, but it's built on a lipid template of 4 main lipids, the same that you can find in chloroplasts, so MGDG, DGDG, PG, and SQDG. The 95% of the prolamellar body membrane proteome is built by the enzyme called protochlorophyllite oxidoreductase. This is the light-dependent enzyme that forms ternary complex with the chlorophyll precursor called protochlorophyllite and also the NADPH. When the light is switched on, the reduction occurs and the protochlorophyllite is reduced to the chlorophyllite. This is the only reaction on the pathway of the chlorophyll biosynthesis that is light dependent. So when there is no light, the plant is arrested at this step. Also inside the membrane structure protein called curd protein can be found, and this is the thing that we lately shown, and I will talk about it more later. And also carotenoids are present directly inside the lipid bilayer. So we asked 2 questions. First of all, what is the exact nanomorphology of the PLB? Because we know that this is the type of the cubic membrane, but there is a lot of different assertions in the literature known from 70s that did not have enough of the evidences to confirm the true structure of the PLB. And also we wanted to know how changes in the PLB composition, namely those components that I listed before, could influence its self-organization. So let me start with the first one. And this first one was in fact not that easy, because to do so, to recognize the nanomorphology of the PLB, we had to work with the new method and invent a new method to work with this. Always the observations of the structures with the nanomorphology and the size of the particular unit cells in this structure is around 80 nanometer, has to be combined with the proper method. In this point, we had to use electron microscopy, because light microscopy has not enough resolution to recognize the structure of the prolamellar body. Just to remind you that in electron microscopy, in the transmission electron microscopy, we always observe a 2D projection of the 3D object. This 3D object is very thin because it's around 90 nanometers thick. And for the lamellar structures, it might be recognized as something that is somehow infinitely thin. But for those very condensed and very periodic structures, even in this very thin sample, we have a lot of 3D structure going on. So we have to remember that what we see here is in fact the projection of the 2D structure. At the beginning, it was always projection directly onto the photographic film, but now it is the projection on the CCD camera, and then the photograph is visualized on the screen of your monitor. When you would like to visualize this structure in 3D, you can use the technique called electron tomography, in which you rotate the sample inside the microscopy chamber and collect the series of slices, the projections at different angles, which are then computed to be parallel, which is the tomogram. Based on this tomogram, you can reconstruct the 3D structure of the sample. Unfortunately, when the structure is very condensed, like in terms of the prolamellar body, this reconstruction is not very precise. So the recognition of the particular type of the mathematical surface that we would like to recognize is somehow impossible. So we developed a new method by making the software called SPIRE, Surface Projection Image Recognition Environment, in the collaboration with the group of Professor Gert Schroeder-Turk from Murdoch University in Australia, and also the group of Smith Evans in Potsdam University in Germany. And what we did is we implemented different types of triply periodic minimal surfaces, and this enabled us to calculate 3D objects based on those templates, but having all of the parameters that can be read directly from the TEM image. And as such, we can calculate in SPIRE the slice, the synthetic slice of the specimen that is put into the microscopic chamber. Then we make the projection of it. So we make a synthetic TEM image. And through the template matching, so through the matching of those two images together, this computed one from SPIRE and the TEM image, we can recognize the surface with a very high accuracy. Obviously, we can also generate slices with the 1 pixel thickness and then make the 3D model of your TEM sample based on only 2D data that were provided from the typical transmission electron microscopy. In SPIRE, you can of course recognize not only structure characteristic for the prolamellar body, but for all cubic membranes based on several different types of triply periodic minimal surfaces like primitive, diamond, or gyroid. You can implement different parameters and calculate 3D parameters based on only on the analysis of the 2D slices. You can calculate the inner volume of this structure, the membrane surface in 3 dimensions, the outer volume also. In terms of prolamellar body, the inner volume is the lumen and the outer volume is stroma. We also add a very interesting parameter, which we call the percolation threshold. And this parameter enabled us to calculate the diameter of the largest sphere that could fit the channel. And this, in terms of the biological membranes, has a tremendous consequences in terms of constraining the molecular motion. So we can ask whether the particular molecule of a particular size could fit or could not fit the structure. So whether it could penetrate or not. And what is the workflow here? So we have to take the image. Here you see the Prolamellar body, and then make the qualitative analysis in which you can separate all of the polycrystal parts of the image. Then you have to make an educated guess. So you have to guess which type of the surface it might be before you start the original matching. You can match all of the parts to have a reliable answer to your question, and then even build a synthetic structure based on those projections. As you can see, the accuracy of this procedure is high. So using SPIRE, we were able to recognize that Prolamellar body, PLB, is a geometrically imbalanced diamond. What is very interesting is that all of the cubic membranes known so far were always balanced. So all of both channels had the same diameter, but here the structure is imbalanced, which might be related to the function of this structure. So to not only check this on one example that you saw before, we checked this in many different plant species. Here you can see some of the examples. We checked the monocots and dicots. We checked the Prolamellar bodies formed inside true leaves, but also inside cotyledons. And we matched those structures with the diamond surface with high accuracy. We were able to recognize that the single unit cell size is quite constant, varying between 73.5 to 82 nanometers, and the volume proportion, so the proportion of those two channels, is also quite constant, from 0.22 to 0.3. So when we already knew that the PLB is in fact the diamond, in contrast to the long-standing assertion that the surface type that is connected with the PLB is a Wurtzite, now we wanted to know whether how changes in the PLB composition can influence its self-organization. So we tested several different components. Now I start with the CURT1 proteins. CURT1 are the transmembrane proteins which were discovered in fully developed thylakoids and connected with the regulation of the structure of the thylakoid network. But it was not known whether they appear in time during the development of the chloroplasts, such as all of the photosynthetic complexes that are not present in etioplasts, or maybe this protein is present already from the very beginning of plastid development. So we tested this and we realized that CURT proteins are present already in etioplasts, and the amount will grow in time during the development on light. So those numbers indicate hours of light. We also observed that lack of these proteins in CURT1 A, B, C, D mutants, so the mutant that lacks all of the proteins from CURT1 family, during the biogenesis, these plants had an impaired greening, so the biogenesis process was arrested. So obviously we were interested how the lack of this protein or the overaccumulation of this protein, which did not influence the process of the chloroplast biogenesis, will influence the structure of the PLB, and results were quite striking. So plants lacking even one protein of the CURT1 family, so the main protein CURT1A, had very fragmented PLBs with a larger size of the unit cell, while the plants over-accumulating CURT1 proteins had very compacted PLBs. And we, of course, tried to recognize whether this compactness could be connected also with the change of the type of the surface from the diamond to primitive of the gyroid. But we realized that even though the structure is getting more and more condensed, the type of the structure is still the D-type surface, even though the volume proportion is changed, but also the unit cell size is significantly decreased. So this is the model. It's always hard to see this on the slide. So I prepared, maybe you can see me. I prepared 3D printed models of the typical slice of the prolamellar body that it resembles the specimen of the TEM sample, and also for this over-accumulating curd mittens. So you can see that the condensation of the structure is much higher. And if you could take them in hand, you could also see that this weighs much more. So this is the thing that has to be mentioned, that in these structures, a lot of membranes can be stored, and the more condensed the structure, the amount of membrane that can be stored increases dramatically. So then we were curious, what if we change the amount of one of the parts of the ternary complex, so this L4 protochlorophyllite NADPH complex. So if you have plants without L4 or without the protochlorophyllite, this is lethal because plants cannot develop chlorophyll upon illumination. So the seedling cannot live for a long time. So we tested the plants over accumulating protochlorophyllite. Here you can see the 77K, so low temperature protochlorophyllite spectra to compare wild type col0 plants with PIF1 mutant, which over accumulates this chlorophyll precursor. What we know from this is that most of the overaccumulation of the protochlorophyllite is not connected with the complex, with the complex of the ternary complex, because this peak corresponds to the ternary complex, but rather it overaccumulates directly in the lipid bilayer. And how it influenced the 3D structure, we observed that the unit cell size wasn't changed, but the volume proportion increased from 0.25 characteristic for Arabidopsis to 0.5. So still, the structure was a diamond, but this time it didn't influence the size of the unit cell, but only influenced the volume proportion of the channels. So we asked ourselves whether it could have any influence on the function of the prolamellar body, which is still not yet discovered in full spectrum. So we know that the PLB is a storage magazine for the mostly for lipids, but we don't know whether this particular structure could have also different functions. So we used this percolation threshold parameter implemented in SPIRE and combined it with the size of the ribosome from the publicly available crystallographic data, and realized that although the ribosome could easily fit into the large channel of the wild type Arabidopsis, but also all of other wild type plants that I showed you before, but it cannot fit in both OA curd and PIF1 mutants. However, those plants during their biogenesis behave completely normal. So we speculate that ribosome localization inside the PLB network is not crucial for its proper functioning. This also is somehow different than the long-standing assertion, because already from 70s, people thought that the size of the large channel of the PLB was limited by the size of the ribosome. So it couldn't be smaller than the size of the ribosome, because it was speculated that the presence of the ribosome inside the PLB is crucial for its functioning, and we think it's not. Okay, so we also checked carotenoids, and in this terms, we tested 4 different mutants with different disturbances in the amount of different types of carotenoids. We did it in 2D and also in 3D, and calculated several different structural parameters to decipher what is changed. So for example, we calculated the diameter of the small channel of the membrane, and we didn't see any particular differences. But when we calculated periodicity of the membrane directly from the 3D data or from SPIRE, so these X's here are data from SPIRE, and those box plots are data from the electron tomography, we observed that these data fit to each other nicely. So we are happy with the development of our method. And also we saw that the most of the changes in the periodicity of the membrane, the size of the unit cell, is in CCR11 mutant, which has a lowered level of lutein and overaccumulates prolycopen and zeta-carotene, and also in ABA16 mutant, which overaccumulates lutein and also has a lack of neoxanthine and violaxanthine. And although the larger size of the unit cell in CCR11 mutant didn't have any particular role for the periodic structure, in ABA16 mutant, we observed local disturbances in the periodic structure, which aims for the stabilizing role of beta-xanthophylls in the formation of the periodic structure of the PLB. So next step is to discover what's going on when the etioplasts reach light. And this is the process in which we can see that paracrystallinity is lost, and in several steps the thylakoid network is formed. And around this topic there was also a lot of literature assertions, also from 70s, which was the gold era for the ultrastructure, in which people claim that the PLB disrupts into vesicles, and then those vesicles connect once again already into the lamellar phase. So we tested this hypothesis, and so to answer how PLB transformed into lamellar arrangements, and wanted to extend this analysis on the level of the first grana formation. So how Granostax grow during these early stages of chloroplast biogenesis. And let me start with the first one. So we tested this on the model plant for us for this experiment, which was the runner bee. So we tested this process in first true leaves, which is always better than tested this in cotyledons. And we checked how the PLB transforms into first grana stacks. We tracked this during 3 days of day-night growth to mimic the natural conditions. At first, we tested all hours, so hour by hour during 3 days of the day-night growth, but then we selected key points to perform the 3D analysis. So first, you can see the reconstruction of the 3D structure of the periodic prolamellar body. And when the light is switched on, immediately, even after 10 minutes, we can see the loss of the paracrystallinity. However, we did not observe any disruption into vesicles, and this structure directly transformed into the lamellar phase. So even at this very, very first step of the chloroplast biogenesis, the structure is already lamellar. Next step, after 2 hours, we observe only remnants of the PLB, and the stroma thylakoids remained porous, which is characteristic for also the stage of the etiolated seedling. The prothylakoids surrounding the PLB are also porous. After 4 hours of illumination, no remnants were observed, and only loosely parallelly arranged thylakoids, porous prothylakoids to the long axis, parallel to the long axis of the plastid. And what we also discovered there, that they start to dichotomically split to form first membrane stacks. And what was very interesting was that even at this very, very beginning of the grana formation, already this structure resembles the structure of fully developed grana in which stroma thylakoid is connected with the grana thylakoids in a staggering way, so in so-called helical way. We also observe how these grana grow during time. So at the beginning of the second day of the experiment, so after night, grana were larger, but still the helical character was maintained. And finally, we also checked how this grana structure will look like after 3 days of illumination of, in fact, day-night growth. And this structure was very similar to the structure after 2 days, but only larger diameter and more layers were visible there. So you can see here that still the helical character is maintained, but the structure become more complicated. So to sum the most important things here, first of all, PLB does not disrupt into vesicles, but rather transform directly into flat slats. The first initiation of stacking is initiated by the dichotomic split of parallelly loosely arranged stroma thylakoids, and then the first grana stacks are formed by already having this initial helical arrangement. So we also wanted to know how those grana stacks that we observed grow during these early stages of chloroplast biogenesis, because we know that in fully developed chloroplasts, the grana are much larger than those first initial steps that we were tracking. So we measured several different grana parameters, like a diameter, height, and number of membranes in the granum stack. And we observed changes during time. First of all, the grana were being more stacked. So the SRD parameter, which is the distance between the adjacent partition gaps in the stack, decreased to reach the values characteristic for fully mature plants. And also we calculated and introduced the parameter that we called grana lateral irregularity, which is a coefficient of variation of membrane diameters with the granum, which means the grana become more regular on time. And also we checked that the tendency of grana membrane expansion is changing from the lateral direction. So grana first reach their diameter and then they build up next layers. We were also interested whether those more and more regular grana distributes more regularly in the chloroplast, what was checked using CLSM method. And we observed that grana were distributed more and more regular during the time of biogenesis. So finally, we stopped our experiments after 3 days, not because the grana reached the size characteristic for fully mature chloroplasts, because those granas have a lot more membranes, typically a lot more membranes, but we stopped this experiment at the level when plants reached the efficiency of the photosynthetic apparatus comparable with fully mature plants, and because we were completely exhausted after 3 days of day-night experiments. So the next step that we are studying is the fully developed chloroplast thylakoid network. And we can observe there, obviously, the typical grana arrangement with stroma thylakoids surrounding this space. Obviously, as I introduced at the beginning, these are in fact flattened vesicles that are interconnected within the continuous way, in the continuous form, which separates inner compartment as a lumen and the outer compartment as a stroma, which is crucial for the light phase of photosynthesis, as everybody knows. So obviously this membrane is built by lipids, the same as in the Prolamellar body, so MGDG, DGDG, SQDG, and PG. And even though we know a lot about membranes of thylakoids, we still don't know whether they are evenly distributed or they are distributed unevenly in different parts of the structure. And still there is a bit of the struggle to decipher this, and there are different opinions in different groups using different methods. However, everyone agrees that proteins are distributed unevenly. So those stacked regions are enriched with photosystem 2, PS2 with its antenna, and those stroma thylakoids and end membranes of grana are enriched with PS1 and its antenna and ATPase, while cytochrome B6F is distributed quite evenly. We have to remember that even though those proteins are about 70% of the mass of whole membrane, we also know that there are curved proteins already mentioned that are inserted in those grana margins, so in these curved regions, because this name of the proteins is from curvature, this curved protein from curvature. And also we've got a lot of carotenoids. Some of those directly inserted into the membrane, but most of them inserted directly into the photosystems. So obviously we know a lot about proteins and a lot of groups studying proteins and mutants with different amounts of proteins. So we wanted to focus on those sometimes omitted elements. So we wanted to know how the galactolipid composition influences grana structure, and then what is the structure of carotenoids in the thylakoid network formation. So let me start with galactolipids. We tested two mutants, the mutant that had a lowered level of DGDG, so this bilayer forming lipid, and the mutant MGD12 with the lowered level of MGDG, which is a non-bilayer forming lipid. So this is a lipid that, if it is not connected with other lipids or proteins, will form inverted hexagonal phases rather than bilayer. So obviously we started with the ultrastructure because this is the center of our interest, and we calculated different parameters. And no surprise, because plants that were lacking some of the lipids had smaller grana with the smaller diameter and smaller height. And also this SRD parameter was this time larger, so more characteristic for the younger seedlings than fully developed seedlings. But we didn't see any particular differences between MGD12 and DGD12 mutant, which was quite surprising for us, because MGDG, due to its structure, has a completely different influence than the DGDG. So we checked this in 3D, and then we realized that there is a huge difference that could be observed only in 3D. So in DGD12 mutant, the structure in 3D was completely identical to the structure of wild type in terms of the type of the connection of the stroma thylakoids to the grana thylakoids. Here is the staggering one, so the calical one, and here also you can see the calical arrangement. While in MGD12 mutant, so the mutant with lowered level of MGDG, we observed changed structure of these regions, which we called calical dichotomic, because still the membrane is staggering, connecting several different layers of the membrane and making the continuum of the lumen, but the membrane dichotomically split and stay connected with the previous layer for a longer area. So we were curious whether such structures can influence somehow the function of the chloroplast, because still we always want to connect the structure with the function. And we realized that even though there were not a lot of changes in the arrangement of the photosynthetic complexes or amount of photosynthetic proteins, and even the efficiency of the photosynthetic apparatus as a whole was similar, we observed that the ratio between cyclic electron transport and linear electron transport was increased in MGD12 mutant compared with DGD12 and wild-type plants. We concluded that most probably those complicated connections in the marginal region can provide a steric hindrance for the transport of the long-range electron carrier, so the plastcyanin, between stacked regions and stroma thylakoids, which is crucial for the proper functioning of the linear electron transport. So our next question regards carotenoids and their structural role. And this situation is even more complicated because carotenoids are both localized directly in the lipid bilayer, but also inside the photosynthetic complexes. So we used 4 different mutants with disturbed amount of carotenoids, with increased amount of lutein, decreased amount of lutein, increased amount of biolexantin, or for example, decreased amount of carotenes, and tested how this will influence different parameters of those plants. So it is a lot is known about the influence of the carotenoids on the photosynthetic process per se, but we wanted to focus on this structural role. But we had to test both the lipid fraction and the protein fraction due to its double localization of carotenoids. First of all, we checked the efficiency of the photosynthetic apparatus, and we realized that probably the most interesting would be those SZL11 and SZL11 NPQ12 mutants. So these mutants over-accumulating lutein together with a decreased amount of carotene. And first, we focused on the lipid phase. We obviously checked the composition of the lipids, so MGDG, DGDG, SQDG, and PG, but not a lot of important changes were found there. But we tested the fluidity of the membrane, since it is known that carotenoids can influence the fluidity of the lipid bilayer, and lutein is known to stiffen the membrane. So we used the fluorescence diode called Laurdan and calculated so-called general polarization value. The larger the value, the membrane is more stiff. And we observed that in case of mutants over-accumulating lutein with decreased amounts of beta and alpha carotene, the membrane was more stiff. So we know that this structure is stiffer. Then, of course, we have to focus on the protein phase. We analyzed the organization of the photosynthetic complexes using blue native phage and also low temperature fluorescence of the chlorophyll bound with different complexes. And we realized that the composition is mostly disturbed still in these 2 mutants that also had a different fluidity of the membrane. Here we recognize higher amount of PS2-LHC2 supercomplexes of low mass, and also the presence of the additional bond, which corresponds with LHC1, LHC2, PS1 complex, what was also confirmed by the 77K fluorescence, where the shift of the peak corresponding to PS1 was observed. So we tested also different proteins and realized that in terms of the amount, there is not a lot of changes. We also tested this in the SDSP gels, where we realized that LHC2 level in general is not changed, which means that the lutein that overaccumulates in these 2 mutants mostly overaccumulates directly in the lipid phase. No surprise, because the phase is more stiff. But we observed something interesting, that with the isolation of the thylakoids co-isolates the stroma protein, so rubisco. And then we, of course, checked the ultrastructure. So the ultrastructure was quite similar in 3D. In all terms, we found the helical arrangement. The diameter was constant in all of the mutants, but what we observed was that in those 2 mutants overaccumulating lutein with lower level of beta-carotene, the number of thylakoid layers was significantly lowered. So compared with wild-type plants, the height of the grana in those LUTCAR mutants was significantly decreased. We conclude that this is a synergistic effect of the lutein that over-accumulates in the thylakoid lipid fraction by stiffening the membrane, providing the hindrance for the folding of the Granostax, and also this stiff membrane connects with those functional rubisco octamers, which could also provide an additional steric hindrance for the process of the folding. And also synergistically, it works with the lower level of carotenes, which influence formation of different types of super complexes. And from other literature data, we know that larger amount of low mass supercomplexes influence also the stiffening of the membrane. So we know that the membrane which is more stiff, which is directly connected with the carotenoids localized in the lipid bilayer and proteins, will influence the granostructure. And as I showed you at the beginning, it also influenced the general efficiency, photosynthetic efficiency of those plants. And finally, last slide here, because we were very focused with carotenoids only on the granostructure, but then we realized that something very interesting can go on in stroma thylakoids. And in mutant that overaccumulates zeaxanthin with the lower level of lutein in fully developed plants, so ABA16 mutant, the same mutant that had this disturbed prolamellar body, it can transform the lamellar phase into the cubic phase. And using SPIRE, we recognized that this cubic phase is of a gyroid type. But so this is not a diamond, this is still thylakoid membranes, but forming completely different arrangement. And also the scale of this structure is completely different. Now I'm showing you this is in scale. So this is the periodicity of the PLB compared with the periodicity of this gyroid structure. And this is the thing that we now still exploring and try to figure out how does it work. So we are checking the composition. We are fractioning those membranes, checking their composition. We also have another model to study these cubic membranes, but this is quite new phenomenon. Until now, we didn't know that thylakoid membranes could even form this type of organization. So this is the thing that we are focusing lately. So to sum up everything, this is the slide that sums up different topics that we are currently working on or are already finished. They all are related in the with the structure of the plastids, so the inner membranes of the plastid, directly connected with the influence of the composition of those membranes on the structure, but also the influence of environmental factors on those structures. But today I was able only to tell you about some of those, about Kurt proteins in etioplasts that modulate the structure in the dosage-dependent manner, and about polar lipids in fully developed chloroplasts that could change the chemical arrangement into chemical dichotomy one, the pigments in etioplasts that could influence the volume proportion in terms of overaccumulation of the protochlorophyllite, and also can destabilize PLD in terms of the lack of beta-xanthophiles, and can influence both the granostructure that could decrease the process of their folding, and also could lead to the completely new type of structural arrangement noticed in fully developed chloroplasts. So I would like to thank all the people who were involved in this work. So postdoc students, Radek Mazur and Michał Bykowski, and PhD students, Joanna, Ania, and my master students, and also all the people who collaborate, with whom I collaborated on those projects, Gert and Myth that I already mentioned, Ralph and Omar with them, and with Matthias, we collaborated on the project of Kurt1 protein, and Jakob and Dainius, who are also involved, who are mostly involved in projects of the influence of light on those processes that I couldn't speak about today due to the time limitation, but they were also connected with this project of the SPIRE software that I introduced in the beginning. And thank you very much for hearing my talk, and I will leave you with my contact data. So if you are interested somehow in structure and would like to contact me, you are very welcome to do so. And I will leave you with this, in my opinion, beautiful, but very non-typical, but to be surprised, very functional chloroplast with the gyroid structure inside. Thank you.
Thank you, Łucja, for this very nice talk. It was very, very interesting and also very didactic. I have many questions, and even several of those you answer a couple of slides after. So now we will open the round of questions, and as we used to do, first I open the questions for the PhD students, master students, postdocs. So yeah, someone want to ask something to Łucja? Okay, so then can you hear me?
Yagish
Yes.
wants to ask.
Okay. Łucja, can you stop the sharing of the
Yes.
screen just for a moment? Because yeah, Jagis, because I couldn't see. Yes, Jagis, please.
You shouldn't forget me. Can you hear me clearly?
Yes.
Okay. This is Yagis speaking from Professor Salim Elbab Ali's lab. So 2 years ago, we did, by the way, thanks for really nice, brilliant presentation. Actually, that made me quite excited and then drive me to ask this question right away. So we did a small scale study 2 years ago, published in eLife. Maybe you're aware about that. So in that study, we found an indication that actually cis-carotenoid apocarotenoid retrograde signals might be involved in regulation of Plb formation and then chloroplast biogenesis in Arabidopsis at least. Do you have any studies about the apocarotenoids at the moment currently ongoing, or do you investigate their role in Plb formation? If yes, could you please explain? If no, do you have would you like to collaborate if if this is possible?
To answer to your question directly, we are not following this path of investigation. So we are not currently investigate the role of apocarotenoids, but in fact, we've got some results not only for CCR11 mutant, but also the CCR12 mutant that was published in the eLife. And we actually observed that in this CCR12 mutant, there are some changes in the shape of the prolamellar body, as you observed in the paper. But we also observed that when we change the nutrition media, so with added sugar or without added sugar, it tremendously influences the structure. So this is the thing that it's probably worth to explore, how those pathways can be redundant in some places. And also the CCR11 mutant accumulates some of the apocarotenoids. I showed that prolip and zeta-carotene are there, but the amounts in CCR11 mutants are lower than in CCR12 because this is like a different source of the mutation. And we only, here in CCR11, we only observe the enlargement of the unit cell, but probably if the enlargement would be even bigger, then the PLB would be disrupted. And this is somehow the example of the ABA16 mutant that I showed, that when it has a very large unit cell, and then there are some partial disturbances in the structure because probably of the lack of the stabilizing role of the beta xanthophiles. But in fact, it might be also directly connected to the fact that the too large unit scale cannot be still so stable, even though still plants are amazing and biological tissues are amazing, because all of the artificially explored lyotropic phases, so such as the cubic phases, but made artificially by physicists and chemists, can reach the size of the unit cell at maximum 10 nanometers, while plants, in terms of this ABA that I showed you at the last slide, it's around 500 nanometers. So this is of a large interest also from the interdisciplinary point of view, because people are very interested how to achieve this synthetically to derive medicines and so on. So I just made some comment, but yeah, we are not studying it now, but obviously we are happy to collaborate on this topic because we know a little bit about it and we got
I would
some.
be so happy if we can do that, because I'm also aware that like the cell size differences and also the impact of the sucrose by bypassing the photomorphogenic development in between the scota and photo actually. So I would love to contact you later through maybe emailing and maybe a Zoom call if possible.
Yeah, sure. Drop me a line and we can arrange the call. Thank you.
Thank you. Thank you.
Okay, thank you, Yagis. Is there someone else with questions for Łucja?
May I ask a question?
Salim, please.
Thank you very much, Łucja. Really very beautiful story. And unfortunately, only few people are working on this important part of biology. We are living in the age of omics. How do you explain this special structure in the ABA1 mutant?
Yeah. So in terms of the fully developed plant, so this gyrate structure.
Yes.
So I showed you this because it's always good to show some results that we are not yet fully understand. So maybe someone will have some ideas. But in fact, we found another model to study this. So we realized that this structure is persistent in the ABA16 mutant, but can be formed also in the mutant that lacks one of the kinases of the thylakoid network called STN7, the one that phosphorylates LHC2 proteins. And this structure can be also found in this STN7 mutant. So we are exploring now all of like using these two models parallelly and trying to figure out what are the key things that leads to this transformation. So we spend a lot of time to tune the procedure of fractionation to have these gyroids isolated, and now we confirmed with the TEM of the isolate that we can isolate it purely. And we are now studying all of the things, not only the composition of the lipids, carotenoids, proteins, their organization, but also we are studying, and we are trying to study because it's somehow hard at this point, to study also the aqueous surrounding. And we think that maybe this aqueous surrounding can be crucial at this point of the investigation, but we are too early at with our results to give you the direct answer.
In the ABA1 mutant, is the the phosphor pattern, is it affected?
Yes, somehow yes, but not that tremendously as in STN7 mutant.
And is there anything known about the regulation of this kinase?
About the regulation of this
kinase,
we know. Yeah, no, this is not touched path, and this is the thing that we would like to follow because we have to connect somehow.
Very interesting. It might be induced by ABA itself or by another signal coming from downstream carotenoids.
Yes.
Which are missing.
Yes.
From structural point of view, it doesn't make sense that if you accumulate zeaxanthin, you would get such a huge modulation of the structure.
So we know that STN7 mutant does not overaccumulate zeaxanthin and still forms those structures. So that's why we don't believe at this point that it is really connected with the zeaxanthin. At the beginning, we thought so because zeaxanthin can really modulate the stiffness of the membrane. We know that the more of the zeaxanthin from the molecular studies, like in vitro studies, the more of the zeaxanthin, the membrane is more stiff. But to, produce the cubic arrangement, the membrane shouldn't be very stiff, so it doesn't make any sense. So we believe at this point that it is not connected with the actually overaccumulation of the carotenoid there, but rather the connection of the ABA pathway with the STN7 pathway. And this is the topic that we try to parallelly explore. But now our main focus is to figure out what's going on in terms of the composition that enables formation of this structure, then to try to model this synthetically, like in silico, and then try to achieve this in model membranes by the mixture of lipids with particular components, because we know that it is like somehow membranes that you can produce from the thylakoid lipids with thylakoid proteins can form something that resembles thylakoids. So we believe that we will be able to achieve this, of course, not in the very, very near future, but the crucial thing for us now is to decipher what's going on and what is crucial there, what connects these two mutants, and it is not as accenting, we know right now.
The other question is, there are all the studies about the role of Psi in the formation of Prolamellar bodies, but I'm not sure.
The phytosynthetase.
Phytosynthetase.
Yes, we didn't explore this topic, so I cannot tell you not more from our results there. But actually, the thing that we are exploring now, but also I didn't have time to talk about this, is how the PLB is formed. Because this is the huge question mark and no one knows. So most of the experiment starts with the level of the PLB and then it's on light transformation. But the question how the PLB forms and what is the sequence of this and whether there are different pathways of this is really interesting also from the synthetic point of view to then mimic those structures. So phytosynthetase, sorry, I cannot help you with this.
No, I'm not sure about the data, therefore I asked you, I'm not sure about the involvement. And there was many studies around this and the role of carotenoids in the formation of the Prolamellar body, push and pull and so on. Okay, thank you very much. Really
Thank
excellent.
you.
Okay, someone else? If not, I would like to ask several questions, or well, okay, I will only ask two and then by email or by Zoom we can discuss Łucja. But following also the same line of Salim's question, because I noticed that, of course, you use different carotenoid mutants, and depending on in which step they are blocked or impaired, of course, you have some accumulations, but also you have drastic phenotypes. You have reduced photosynthesis in the two most important mutants that you analyze. So the SZL1 and then the double mutant with the MPQ. You have different phenotype, leaf shape, etc. So I think a better, or what could be, I think, a better approach would be to use overexpressors, which generate or produce the accumulation of different carotenoids. Particularly, well, I generate one of those which produce not only more lutein and zeaxanthin, beta carotene, but interestingly also produce more ABA. So this could be also another layer for your research to confirm or to draw stronger conclusions about what you are concluding with these mutants, right?
Yeah, I completely agree. We just didn't have any over-accumulating plants, so we couldn't test this hypothesis.
Yes.
And we couldn't test this, but I hope that we will.
Because it's very interesting also what you mentioned, that the protochlorophyllide is key for the formation of the membrane and so on. Do you think, because you use the mutant that is over-accumulating this protochlorophyllida, did you check other mutants or overexpressors of the chlorophyll biosynthetic pathway, and you analyze
So no, we only checked the mutant over accumulating protochlorophylite as this because our goal at this point was to check the composition of this membrane and then change only most precisely as we can those components that are already there in the fully developed PLB. So I didn't show this today, but we also checked the mutants with the disturbed levels of lipids, so MGDG and DGDG on the level of the PLB. But surprisingly, even though the changes were quite striking, the structure was almost almostly the same.
Okay.
So, and on the other hand, when you get rid of the pore or protochlorophyllites, so you've got like those mutants without, they are little, so nothing to study. So there is, so we only checked the protochlorophylite, but in fact, it would be nice to check also the mutants that are somewhere there behind in the pathway.
Yeah.
So because for chlorina mutants, so the mutants that have impaired chlorophyll production, we checked, but there was no change in the structure because like we are earlier than the chlorophyll. So so the regulation is not switched on at this point in the darkness, but we checked. We checked the 4 chlorina mutants and the PLB was completely normal.
Because I think maybe the mutants can be a bit more noisy and it will be hard to pinpoint something. But at the moment you include the overexpressors, you can combine both and draw very strong conclusions. And interestingly, so at least in tomato and in tobacco, lines that are overexpressing the lycopene beta cyclase, they also have higher lipid content besides the higher carotenoid content. And in tobacco, the lines I was telling you, they also have higher chlorophyll content. So
we can expect that the grana will be huge.
Yeah, I think so. Something I can tell you is that also in those overexpressors, they have higher chloroplast number. I don't know why, might be related with ABA. Some previous studies show something, but yeah, we don't know. So that will be very interesting to see. Okay, and I would like to make the last question more general, more, let's say, philosophical. Maybe you already got it before, or maybe even you are working in something related. How do you think your research could contribute to artificial photosynthesis or to synthetic chloroplasts, for example?
So actually, this was when I was starting to work with the ultrastructure. This was somehow my goal. Like the goal that I was thinking that, okay, I work in this basic science, but maybe in the future, those results will be helpful for producing something that is artificial and more efficient. And we know already that the structure was very neglected for several decades now. So the 70s was a burst of the ultrastructure, and then we switched to the genetics and then to the omics. But now I realize that we are coming back to this structure, and there is more of the interest nowadays because we now appreciate that the structure itself can have a function. What I showed today was only few examples. For example, those steric hindrance for the transport of some molecules by the different structure in the grana margins. Yeah. But also we know that the size of the grana is really important for the absorption of the light itself. So it is one of the elements, not only the photosystems and the molecular composition of the super and mega complexes, which was very highlighted for many years. Now we know, for now only from the modeling, but we try to figure out some models to study this also in the microscopy, that if the grana is wider or smaller or larger or less regular, then the light is absorbed in the completely different way, more efficient or less efficient. So in these terms, I'm quite convinced that this is another step, that it won't be the first step in artificial photosynthesis, but it will be the next one. So the first would be to produce efficient proteins, but then you have to put those proteins somewhere. So this is the thing. And this is the part of the grana, to answer to your question. But another path of my interest in terms of more applicational use is those cubic membranes. So the PLB or the gyroid. So this is the thing that really I see that a lot of physicists coming to me with some questions. So do you know how exactly is the composition? Because we would like to try to achieve this synthetically. Because if you would like to, we already know that those cubic phases are crucial, for example, in mammal's milk. So when the child is drinking the mother's milk, then those cubic structures are forming inside those milk, and they are opening in the different pH to release the vitamins, antibodies in the good part of the digestion tract. So people are thinking that, okay, so maybe we can do delivery systems for the target medicine delivery, like a drug delivery systems, built of cubic membranes, because they could open in the particular place, in the particular pH, depends on their composition. But the problem is that those molecules of chemotherapeutics are huge, and they cannot fit in those small synthetic membranes that can be achieved by now. So the people would like to inspire with nature, how nature does such huge islets of the network, but it is still stable, to make it synthetically and to use it not only in drug delivery, but also a flavor delivery. Yes. In food industry. So their applications are very, very, very large. So yeah, so this is like with the grana for the artificial, and with the cubic membranes for more of the industrial use. But still, I think that at some points, even though it might seem that these are very basic studies and looking at beautiful pictures, and most of the time, it still have something behind that could be used by a lot of people.
Yes. Okay, then thank you so much, Łucja, for this very, very nice talk. And yeah, we keep in touch.
Thank you. If everyone who is joining and didn't have questions will have questions in the future, I would be happy to answer via email. Thank you.
Thank you so much.
Thank you very much, Łucja.
Bye-bye.
Thank you. Bye.